On this week's Arabidopsis Research Roundup, I'm again delighted to be joined by Dr. Ian Henderson from the University of Cambridge. We're going to be discussing a paper just published in Genes and Development, the title of which is Natural Variation and Dosage of the High 10 Meiotic E3 Ligase Control Arabidopsis Crossover Recombination. So thanks again, Ian, for joining us. Ian joined us on the very first one of these Research Roundup chats. And it's obviously a fundamentally important paper for the way that variation is maintained within a population. So it'd be cool if you could give us a bit of a background and then discuss the paper.
Yeah, it's a pleasure again to talk to you about our research. And our lab works on recombination. And I think one of the real strengths of Arabidopsis has been a great deal of resources and tools built around natural variation in the species. And so one thing we were interested to look at a number of years ago now is whether different populations of Arabidopsis would have different levels of recombination. Now, Arabidopsis is a selfer, but it does outcross. So, we think it is important in the population biology of the species. And certainly when we've looked, actually, we have found a lot of variation in levels of recombination between Arabidopsis from different parts of the world. Mm-hmm. And so basically in this paper what we did was to actually map one of the genetic loci influencing that. It turned out the gene itself, as you mentioned, high 10, was already known to be important. But this is the first time we could show that just in natural populations there was variation in this gene, and that had consequences on basically how much recombination or how diverse the populations could be. Okay. I think where it was also quite surprising was, we were working on this gene, and one of the things we did was transformed back in additional copies. Mm-hmm. And the really surprising result was that then basically put recombination levels even much higher than the wild type levels. Okay. And it turns out that high 10 is very dosage sensitive, so that the levels of this factor, if you go below wild type levels, the recombination drops. But equally, if you put extra high 10 into the system, mm-hmm. Recombination increases. And basically, that's potentially there could be some applied significance there in terms of whether a similar strategy might work in crops. Mm-hmm. Is something we're following up with at the moment.
So introducing high 10 into particular parts of the genome or into different genomes which don't recombine as much, you could increase the amount of recombination, increase the possibility of adapting to different conditions.
Yeah, I mean certainly in many crops like wheat, the recombination levels can be quite limiting to breeders. Mm-hmm. And so it would be, potentially a way that breeders could, try and modify recombination in order to kind of improve that process, the breeding process. Okay. It remains to be seen. It certainly works very well in Arabidopsis, but as you know, the Arabidopsis genome is quite different to something like wheat. So, yeah. We have to go on.
Well, you've got to start somewhere, right? That's the whole point. Yeah. So Ian, can you take us back to the start of the study, really? So you use Columbia and Landsberg ecotypes. Can you give a brief explanation of the technique you used to identify these QTLs, which altered in recombination?
Yeah, that's right. Columbia and Landsberg are the sort of standard laboratory accessions. They're what we call rapid cyclers, so they have a short generation time, which makes them useful for genetics. Mm-hmm. And we've had a number of long-standing collaborations with people in the field who have generated fluorescent lines that allow one to very easily measure recombination levels in individual plants. So our collaborators on that, Greg Copenhaver from Chapel Hill, in the USA and North Carolina, and Avi Levy from Israel.
Right.
And so they kindly provided us these lines many years ago. And the key thing there is that it allows very high throughput measurements. So basically we collect the seed, put it under a fluorescence microscope, and then use image analysis software to count the patterns of fluorescence in that seed or also the pollen. And basically those patterns, from those patterns we can deduce how much recombination happened.
Okay.
And and all of those lines were originally generated in Colombia, and what we found is that when you cross those with Landsberg, you can see these modifier effects.
Okay.
So they do exist. We like to think that they have some importance in the sort of evolutionary history of the species. I think that's a much more difficult question to get to grips with. But again, it is something we're interested in as well.
Absolutely. And these lines rely on the fact that you have a fluorescent transgene, which is a known position within a chromosome, and then introducing, obviously, a non-fluorescent chromosome, you can track where those transgenes end up in subsequent generations. Yeah,
that's exactly right.
Okay, cool.
So it's quite simple, sort of genetic principles. But like many things in biology, the availability of fluorescent proteins of different colours just really facilitates the work. It would be extremely laborious to do this without those systems. So
absolutely.
They were very clever things that the collaborators I mentioned built over several years.
Cool. So I have a final question for you about the actual protein itself, the high-10 protein. So it's an E3 ligase. So so you said a bit of work has been done on it previously. Do we know anything about the targets for that ligase?
I think that's very much an area of active research. It turns out it's a very conserved protein, so you find it in humans, in plants, like many of these recombination proteins are very conserved. And it's thought to basically transfer ubiquitin groups to factors involved in recombination. Now, it's not, to my knowledge, known what those targets are, so like Arabidopsis, but there are some good candidates. We assume that they're some of the key players are modified by high-10, and that then changes the chance of a recombination event happening.
Yeah,
but it's quite, in the work we do show is quite special. Like other genes in the same pathway don't have the same dosage sensitivity. So it really is, there's something special about high-10 and what it's doing. Again, we don't understand what it is biochemically that causes that distinction, but it's a very sensitive point in the pathway.
Yeah, no, I imagine that would be an interesting relationship, because you imagine if it was very promiscuous protein, then it would modify lots of different proteins, and it doesn't matter how much was there, but there might be a more one-to-one relationship with whichever target it is. So it would be interesting to find out ultimately what those targets are, certainly. Yeah.
I mean, one curious thing that came out of it is if you go and look at high-10, well, in humans it's a bit more complicated. There's another protein called RNF212 that's kind of related to high-10, thought to act in a somewhat similar way, so also controls recombination. And when there was a study in the Icelandic population looking at variation in recombination between human families, and RNF212 was identified in that study as. So it's kind of intriguing that the same gene or family of genes you would find associated with natural recombination levels in very diverse organisms like plants and humans. My interpretation of that, again, is that reflects the fact that these things are so dosage sensitive that they crop up again and again for that reason.
Yeah, it's fascinating. Absolutely. Great. All right, well, Well, thanks very much, Ian, for chatting to us today. And I remind listeners that Ian was one of the prime organisers of a GARNet meeting on natural variation that we had at the end of last year. And if you go to the GARNet website, you can get the abstract book from that meeting, which went quite well, I think. But obviously things are going very well for you, Ian, in Cambridge there. So we look forward to hearing about your future developments in the future, obviously.
Well, thank you very much.